For decades, the conversation surrounding Alzheimer's disease has relied really heavily on the terminology of a military campaign.
Oh, absolutely.
Right. The headlines always focus on, like, launching an attack against the disease or targeting the amyloid plaques or going to war against the symptoms. We've basically treated the brain as this static battlefield where neurons are just, well, slowly losing ground.
Which is an approach that fundamentally ignores the actual biomechanical environment of the brain. I mean, when you frame the disease as a direct assault on the pathology itself, you miss the structural systems that allowed the pathology to accumulate in the first place.
Exactly. And that brings us to the mission for today's custom-tailored deep dive for you listening. Okay, let's unpack this. We have a genuinely fascinating piece of source material today from May 2026.
Yeah, it's a great one.
It's a Science Daily article detailing some incredible research out of the journal Signal Transduction and Targeted Therapy. Basically, an international team from the Institute for Bioengineering of Catalonia and Sichuan University has successfully reversed Alzheimer's-like symptoms in mice.
Right, but the key is how they did it.
Yeah, they didn't do it by attacking the plaques with traditional chemical drugs. They used a radically new kind of nanotechnology to essentially repair the brain's internal plumbing.
It is a massive paradigm shift. It moves us away from asking, how do we destroy the amyloid plaque, to asking, well, why did the brain's natural waste disposal system stop clearing this plaque on its own?
Yeah.
And how do we reset that specific machinery?
Right. But before we get into the nanotechnology itself,
Right.
we really have to look at the baseline infrastructure of the brain. Because anyone who follows neurobiology knows the brain is an absolute energy hog.
Oh, yeah.
Even though it accounts for maybe, 2% an adult's body weight, it commands roughly 20% of the body's total energy supply.
Yeah, 20%. And for children, that demand can actually spike to an astonishing 60%.
60%. That is wild.
The metabolic overhead is just astronomical. And it's important to understand what that energy is actually doing. I mean, it's not just fueling conscious thought, right? It's powering the resting potential of billions of neurons and, crucially, maintaining the vast, complex vascular network that's required to deliver all that oxygen and glucose. We are talking about roughly 1 billion capillaries packed inside the human skull.
A billion. The density is difficult to even conceptualize. It means almost every single neuron in the brain is, directly tethered to its own microscopic blood supply.
Precisely. And wrapping around all of those billion capillaries is the blood-brain barrier. Now, historically, the blood-brain barrier was modeled essentially as a passive brick wall.
Like a filter.
Right. A highly restrictive sieve designed purely to keep toxins and pathogens in the bloodstream from leaking into the delicate neural tissue.
But the sources highlight that viewing it as just a passive shield is entirely inadequate, right? It's highly active.
Very active. It operates much more like an intricate system of molecular pumps. It is a dynamic, tightly regulated efflux system. The cells forming the blood-brain barrier expend a massive amount of ATP cellular energy to actively grab waste products from the brain side and pump them out into the blood.
So it's basically hauling garbage out just as continuously as it is blocking toxins from coming in.
Exactly.
It really makes me think of the brain as a massive, hyperactive city. And for you listening, imagine a city that consumes that much energy. It's going to produce a massive amount of trash. The blood-brain barrier is basically the city's sanitation department, making sure the garbage gets hauled away before it spills into the streets.
That's a great way to look at it.
Which brings us to the specific waste product at the center of Alzheimer's, right?
Okay.
Amyloid beta. We all know amyloid beta is the sticky protein that accumulates and forms those infamous plaques in Alzheimer's patients.
Right, the classic hallmark of the disease.
But if the blood-brain barrier is this highly efficient, active pumping system, why is the amyloid beta piling up in the first place? When does the sanitation department go on strike?
Well, the researchers pinpointed the failure to a highly specific piece of molecular machinery. It's a transport protein called LRP1.
LRP1, okay.
Yeah. You can find LRP1 on the surface of the endothelial cells that actually make up the blood-brain barrier. Under healthy conditions, LRP1 acts as a scavenger receptor.
Meaning it goes out looking for the trash.
Exactly. It recognizes the amyloid beta floating in the brain fluid, binds to it, pulls it through the cell, and releases it into the bloodstream for the body to filter out.
So LRP1 is the actual cargo mechanism.
Yeah.
But the sources describe a breakdown in this process that hinges on something called binding affinity. It's a delicate balance, right?
Extremely delicate. The binding interaction between the LLP1 transport protein and the amyloid beta waste operates on a very strict gradient. If the receptor binds to the amyloid beta too weakly, it obviously can't secure the payload.
So the waste just isn't removed.
Right, it slips right out of its grasp. But the more insidious problem seems to be when it binds too strongly.
Too strongly.
Yes. If the binding affinity becomes too high, the LRP1 protein essentially grabs the amyloid beta, undergoes the cellular transit to the blood side of the barrier, but then, well, it refuses to let go. It holds onto the waste. Because the receptor is now occupied and stuck, the entire transport machinery jams. The receptor can't cycle back to pick up more waste, and the whole system breaks down.
Okay, that reminds me of like a magnetic crane operating in a scrapyard.
Oh, sure.
Right. If the electromagnet on the crane is running too weak, it tries to pick up the scrap metal, but it just falls back onto the pile.
Yeah.
But if that magnet is running too hot, if the current is too strong, it picks up a massive load of scrap, swings it over to the furnace, and then physically cannot drop the load.
Yes, exactly.
The crane just sits there, completely paralyzed, holding a pile of junk, and the whole scrapyard backs up.
That is a highly accurate way to visualize the cellular mechanics at play here. And what's fascinating here is how this specific failure drives the paradigm shift we mentioned earlier.
Right.
For decades, the focus has been on the neurons. The neurons are dying, so how do we save them? But this research heavily reinforces the vascular hypothesis of Alzheimer.
So it's an infrastructure problem.
Exactly. The disease isn't primarily a neurological failure initially. It is heavily driven by this underlying infrastructural breakdown. The blood-brain barrier stops pumping, the magnetic cranes get jammed, and the subsequent toxic pileup is what ultimately chokes the neurons.
So if the LRP1 receptors are jammed, I guess the old instinct would be to flush the brain with traditional chemical drugs to somehow dissolve the plaque. But that obviously hasn't worked effectively. So how did the researchers from IBES and Sichuan University fix the jammed cranes?
They turned to nanotechnology, specifically a process known as bottom-up molecular engineering, to create what they call supramolecular nanoparticles.
Supramolecular.
Right. They built these particles molecule by molecule, achieving immense precision over their size, their physical shape, and the specific arrangement of surface ligands.
Which are basically what?
They're essentially chemical keys protruding from the outside of the particle.
Okay, here's where it gets really interesting, because I want to push back on this terminology a bit. I'm looking at this supramolecular concept in the source and struggling to see the mechanism.
How so?
Well, when we usually discuss nanomedicine, the nanoparticle is just a hollow biological FedEx truck, right? It's a delivery vehicle that crosses the blood-brain barrier and drops off a payload of chemical medicine.
Right, the Trojan horse approach.
Exactly. But the source indicates these supramolecular nanoparticles don't have a payload. How does a hollow physical object act as a drug to solve a chemical binding problem?
That is the crucial distinction of supramolecular chemistry. There is no traditional active pharmaceutical ingredient hiding inside.
Wait, really?
Really? The physical nanoparticle is the therapy. Supramolecular chemistry relies on non-covalent interactions, things like hydrogen bonds and van der Waals forces.
So it's about shape and proximity.
Exactly. Because these nanoparticles are engineered with such a precise spatial arrangement of those ligands on their surface, they don't just randomly bump into the LRP1 receptors, they physically interact with them in a highly choreographed way.
So they are engaging the receptors directly. But how does that fix the binding affinity? Like, how does it make the crane drop the scrap metal?
Through conformational changes. When the supramolecular nanoparticle encounters the jammed LRP1 receptors on the blood-brain barrier, its specific shape and surface chemistry allow it to bind to those receptors and physically alter their three-dimensional structure.
It bends them.
It changes the mechanical tension of the protein, yeah. By shifting the receptor's conformation, the nanoparticle basically forces the LRP1 to loosen its molecular grip.
Oh, wow. So it acts as a voltage regulator for the magnet.
Precisely. It recalibrates the binding affinity back to normal levels. The LRP1 receptor drops the amyloid beta into the bloodstream, the receptor resets, and the cell can immediately cycle it back to the brain side to pick up another load.
That is brilliant.
They aren't destroying the plaque with chemicals, they are physically resetting the machinery so the brain can clear the plaque itself.
Which naturally moves us from the theoretical mechanics to the actual biological testing. And this is where the results are kind of mind-blowing. The researchers applied this to genetically engineered mice.
Right. Mice specifically bred to overproduce amyloid beta and model the progressive cognitive decline seen in human Alzheimer's patients.
And the dosing schedule here really stood out to me. They didn't put these mice on a grueling daily intravenous regimen for months on end. They administered exactly 3 doses of these nanoparticles.
Just 3 doses. And the pharmacokinetics of what happened next were remarkable. Junyan Chen, one of the lead researchers on the paper, reported that within just 1 hour of the injection, yes, within 1 hour, they observed a 50 to 60% reduction in amyloid beta levels in the brain.
Okay, a 50% reduction in 1 hour completely upends the timeline we usually associate with Alzheimer's progression.
Yeah.
I mean, we're talking about a disease where this plaque takes years, sometimes decades, to slowly accumulate and harden.
Right.
How is the brain physiologically capable of clearing half of it away in 60 minutes? It almost sounds dangerously fast.
Well, it speaks to the immense capacity of the blood-brain barrier when it is operating at its theoretical maximum. Remember, the gradient to move the waste out of the brain is already there.
The system wants to clear it.
Exactly. The blood-brain barrier wants to clear it. It has the energy reserves to clear it. The only thing stopping it was those jammed receptors. The moment those supramolecular nanoparticles recalibrate the LRP1 proteins, you are essentially unjamming a massive multi-lane highway. The backlog clears incredibly rapidly because the underlying infrastructure was originally built to handle massive volumes of metabolic waste.
Unjamming the highway is one thing, but clearing the physical waste doesn't automatically mean the cognitive damage is undone, right? Like if the neurons have been subjected to a toxic environment for that long, removing the plaque doesn't necessarily bring them back to life.
You would think so, but the long-term data they collected is staggering.
Yes, the longitudinal study. So they treated a 12-month-old mouse, which, in the context of these specific animal models, is roughly equivalent to a 60-year-old human, an organism deeply into the cognitive decline phase.
Right. And they evaluated that same mouse 6 months later, so the mouse is now 18 months old. It is essentially a 90-year-old human equivalent.
And you would expect a 90-year-old with an aggressive Alzheimer's mutation to be completely devastated by the disease.
Totally.
But after receiving those 3 doses of nanoparticles 6 months prior, this elderly mouse behaved completely normally. It was passing memory assessments, navigating spatial tests, showing 0 signs of the expected Alzheimer's-related decline. How does resetting the plumbing restore lost cognitive function?
This is where Giuseppe Battaglia, the lead researcher from the Molecular Bionics Group, introduces the concept of the cascade.
The cascade.
Right. The pathology of Alzheimer's is a cascading failure. The amyloid beta builds up, which causes inflammation, which damages the blood vessels, which reduces oxygen flow, which then causes more amyloid buildup. It's a vicious feedback loop.
But the neurons themselves might not be dead. They might just be dormant or heavily suppressed by that toxic environment.
Exactly. The brain retains a remarkable amount of synaptic plasticity if you give it a chance. By intervening at the vascular level and restoring the blood-brain barrier first, the nanoparticles effectively reverse the cascade. Once the vascular system is functioning normally, it clears the toxic molecules, the inflammation subsides, and the natural physiological balance of the brain ecosystem is restored. The neurons that were struggling under all that metabolic stress are suddenly in a clean, oxygen-rich environment again, allowing cognitive function to rebound.
That reframing is so vital for you listening right now. Everyone knows someone whose family has been impacted by neurodegenerative disease. So what does this all mean? When we look at how this changes the landscape of treatment, the shift is profound.
It really is.
It suggests that instead of viewing the brain as a collection of isolated neurons that need to be artificially stimulated or protected, we need to treat the brain as an interconnected vascular ecosystem.
And if we connect this to the bigger picture, this approach doesn't necessarily invalidate the other therapeutic avenues being explored. The sources point out that this supramolecular technology could actually operate synergistically with existing treatments.
Oh, you mean like the anti-amyloid monoclonal antibody drugs that have been dominating the news recently?
Yes. The current generation of antibody therapies attempts to bind to the plaque and signal the immune system to clear it. But they face a massive logistical hurdle, which is the blood-brain barrier itself.
Right, because it's still doing its job.
Exactly, because the barrier is doing its job of keeping large foreign molecules out of the brain. Only a microscopic fraction, sometimes less than 0.1% of the injected antibody actually makes it into the neural tissue.
Which means patients have to receive massive continuous intravenous doses just to get a tiny amount of the drug to the target, which often triggers severe side effects, right? Like brain swelling and microhemorrhages.
Correct. The medical field is currently trying to brute force its way past the blood-brain barrier.
Yep.
We have experimental trials using focused ultrasound to literally vibrate the barrier open temporarily, or engineering complex brain shuttle molecules to try and drag the antibodies
That
across.
sounds intense.
It is, but the supramolecular approach bypasses that entirely. It doesn't fight the barrier, it fixes the barrier.
But as we bring this back to the broader picture, we have to look critically at the timeline. Lorena Ruiz Perez, another key researcher on this project, rightfully highlights the undeniable success of reversing the pathology in these trials. The biological mechanism is sound, and the results are incredibly striking.
Yeah, but this raises an important question, right? The translational gap.
Right.
The human hurdle. It is the most sobering reality in neurodegenerative research. We have cured Alzheimer's in mice hundreds of times over the last 20 years. But a mouse brain is fundamentally different from a human brain.
The ratios of glial cells are different, the metabolic rates are different, and perhaps most importantly, the disease models are different. I mean, these mice are genetically engineered to overproduce amyloid beta rapidly, whereas human Alzheimer's is usually a sporadic, complex metabolic failure that develops over 70 or 80 years.
Right. And translating a highly engineered bottom-up supramolecular nanoparticle from a controlled murine model into a safe, reliable therapeutic for humans will require years, likely a decade or more of rigorous clinical trials.
We have to make sure it's safe.
Absolutely. We have to ensure that recalibrating the LRP1 receptors in a human doesn't accidentally trigger a different cascading failure elsewhere in the body.
It's an essential reality check. We can be fascinated by the mechanism and the breakthrough while fully respecting the long, arduous road of clinical validation ahead. So to distill this deep dive down for you, we started by looking past the neurons and examining the brain's immense, energy-hungry network of 1 billion capillaries. We explored how the blood-brain barrier isn't just a wall, but a vital active sanitation department constantly pumping waste out of the brain.
And we saw how that specific transport protein, LRP1, can suffer from a binding affinity failure.
Right, like a magnetic crane jammed with a heavy load, causing sticky amyloid beta to pile up. And finally, we looked at how researchers are utilizing supramolecular chemistry to build precise nanoparticles that act as a microscopic voltage regulator, structurally resetting those LRP1 receptors and allowing the brain's vascular ecosystem to rapidly heal itself.
It's basically a masterclass in treating the root cause of an infrastructural failure, rather than just endlessly battling the downstream symptoms.
And hopefully, the real value here is that the next time you are reading a new study or seeing a headline about a breakthrough in neurodegenerative disease, you'll have a new lens to view it through. You'll know to look closely at the mechanism. Are they just trying to drop a chemical bomb on the plaque? Or are they addressing the intricate underlying plumbing that allowed the disease to take hold in the first place?
It changes how you look at all of it.
It really does. But before we wrap up, I want to leave you with a slightly provocative thought to explore on your own. We spent this time discussing how scientists can essentially reboot a degraded biological infrastructure, the blood-brain barrier, using carefully engineered supramolecular interactions. If we zoom out, many biologists argue that aging itself is, in large part, just a slow degradation of our internal plumbing and cellular transport systems. If it is possible to build a nanoparticle that physically recalibrates the molecular garbage disposal in the brain, could we eventually map and engineer similar supramolecular reset buttons for the rest of our vital organs?
That is a wild thought.
Right. If we can fix the transport proteins in the blood-brain barrier, could we do the exact same thing for the filtration units in the kidneys or the metabolic pathways in the liver? Maybe the future of maintaining the human body won't involve fighting individual diseases at all. Maybe we'll look a lot more like maintaining a classic car. Precisely tuning the valves, ensuring the transport systems are humming, and making sure the underlying machinery is simply never allowed to rust.